Hi everyone and welcome to Unit 9. In Unit 8 we went ahead and talked about our beta and alpha hemolytic streps, and now we're going to get into the gamma hemolytic streps. And some of the organisms we're going to talk about today is Enterococcus species. Of course, that's a gamma hemolytic streptococcus of the gut. And then we're also going to talk about those crazy miscellaneous aerobic gram-positive cocci. Now I would highly recommend that you make your own flow chart for lab. There is a nice one in your book as well, so go ahead and look that up. Let's start by talking about Enterococcus species, and there are several species including Enterococcus faecalis, Enterococcus faecium, Enterococcus durans, Enterococcus avium, and then there are also more species than that. The only ones that we're really going to talk about too much in our laboratory and lecture would be Enterococcus faecalis and Enterococcus durans. Okay, some of these are beta hemolytic and some of them are gamma hemolytic. It just depends on the species that we're talking about. But most of them are considered gamma hemolytic streps. They're no longer part of the group D streptococcus. A pretty good rule of thumb is the only time that we serotype or do the Lancefield serotyping is on the beta hemolytic streps. Out of all the Enterococcus, Enterococcus faecalis is the most commonly isolated Enterococcus, and they are the least fastidious of all the streptococcus. They actually tolerate bile esculin and salt. Most gram-positive cocci are inhibited by bile salts, but Enterococcus can tolerate bile esculin and salt. Okay, they also are aerobic, and there is no carbon dioxide that's needed for their growth, so they're not fastidious at all. Usually they're catalase negative. However, they can be a weak catalase positive if they are very young. If they are less than 24 hours old, they can be very weakly catalase positive. Now, this can be a problem because we don't want to confuse them with Staphylococcus species. The Enterococcus are normal flora of the gastrointestinal tract. There can be a few colonies that are normal in the genital tract as well, but you do not want a pure culture of Enterococcus in the genital tract. Enterococcus, of course, comes from the gut, so if you have Enterococcus in the genital tract, it may be that it's not normal, and it actually comes from stool contamination. Enterococcus can be a pathogen in many circumstances. It can actually cause urinary tract infections. It can cause septicemia. In fact, it's the third most common cause of nosocomial bacteremia. It can cause endocarditis and wound infections, including brain and lung abscesses. And it can also cause genital and respiratory tract infections as well. The virulence factors of Enterococcus species are not well understood. They do produce cytolysins, which are like lytic enzymes, hyaluronidase and gelatinase, and they also produce extracellular surface proteins such as teichoic acids, which are surface proteins or antigens that Lancefield type them as group D. But again, a good rule of thumb is that we don't Lancefield type anything that isn't a beta hemolytic strep. Let's go ahead and start by talking about Enterococcus faecalis, which is the one we're going to be working with in the laboratory. Okay, it is a gamma hemolytic organism on blood agar. Sometimes it looks slightly beta hemolytic on PEA, but we don't really often evaluate hemolysis on PEA. We only evaluate hemolysis on blood agar. Okay, they can be small to medium colonies, and they have slightly a baby blue hue to them, and you can kind of see that when you compare Enterococcus faecalis to some of your Staphylococcus, you can tell that they have a slight baby blue color. They're catalase negative in older colonies, but they can be catalase positive if they are less than 24 hours old, and that's a weak catalase positive. Okay, PYR is positive. Bile esculin is positive, and they will tolerate 6.5% sodium chloride. The CAMP tests are negative. The only reason we would even do the CAMP test is we want to rule out our gamma hemolytic group B strep. Remember, it is important to speciate Enterococcus for correct antibiotic choice when treating infections. We want to make sure that we're not dealing with a vanco-resistant Enterococcus, or if we're dealing with a non-vanco-resistant Enterococcus. We just want to make sure that our organism is susceptible to the aminoglycosides. So let's take a look at a picture of Enterococcus faecalis in the next slide. So Enterococcus faecalis is a gamma hemolytic organism. They are medium-sized colonies, and they have a baby blue hue, which you can kind of see in that bottom image on the right. We try not to confuse it with Staphylococcus because they can be opaque, more opaque than other Streptococcus. Just like our Staphylococcus, we're starting to see resistance in our Streptococcus as well, and that brings us to vanco-resistant Enterococcus, also known as VRE. Most gram-positive cocci, or gram-positive organisms, are sensitive to vancomycin, but we're starting to see some resistance in Staphylococcus aureus and also Enterococcus. So right now I'm going to go ahead and talk about VRE, the vancomycin-resistant Enterococcus. And a lot of times the Enterococcus faecalis and Enterococcus faecium species are the ones that are vanco-resistant. Like I said, most gram-positive organisms are sensitive to vanco, but in 1994 there emerged resistant strains like vanco-resistant Enterococcus. Other countries started to notice this in the mid-80s, but we didn't start seeing this in the United States until the 1990s. Enterococcus suspected of being a vanco-resistant must be tested to make sure they are in fact vanco-resistant. And we can do that by taking Enterococcus and putting it on a bile-esculent azide agar with 6 micrograms per liter of vancomycin. If our organism grows in the presence of milligrams per liter of vancomycin, then it means that we're dealing with a VRE. We could also take our Enterococcus and put it on a brain-heart infusion agar with 6 micrograms per mil of vanco. And if the colonies grow on the BHI with vancomycin, then you know that you're dealing with a vanco-resistant Enterococcus. So let's go ahead and talk about the miscellaneous aerobic gram-positive cocci. The first one I want to talk about is Abidotrophia granulocutella species. I want to talk about Enterococcus, Gamella, and Leuconostoc. Abidotrophia granulocutella species used to be known as a nutritionally deficient Streptococcus, but the new name, of course, is Abidotrophia granulocutella species. And you can read more about this in your textbook, okay? Now, this organism is very interesting. The genetic sequencing has determined that these organisms actually don't belong to the Streptococcus genus, but they are miscellaneous aerobic gram-positive cocci. They can cause things like meningitis and bacteremia following neurosurgical procedures, and they may also cause other infectious diseases like endocarditis. Okay? These types of organisms are gram-positive cocci in pairs and chains. They can be gram variable at times and pleomorphic. And pleomorphic just means that there are different shapes and sizes of the organism in a pure culture. They can be normal upper respiratory flora and normal in the gastrointestinal tract. Like Enterococcus, they are PYR positive and LAP positive. Okay? They can act like some Haemophilus species where they actually satellite around Staph aureus, but they are actually a gram-positive cocci and not a gram-negative rod. And what I mean by satelliting is you can have a colony of Staphylococcus aureus on your plate, and if you have the Abidotrophia on the plate as well, it's actually going to grow around that Staphylococcus aureus. These organisms resemble alpha hemolytic Streptococcus. They're gram-positive cocci and they could be pleomorphic. They're catalase negative like Streptococcus, PYR and LAP positive, and we can determine their vancomycin susceptibility. They should actually be sensitive to vancomycin. Now, other organisms like Leuconostoc and Pediococcus are vancomycin resistant. All other gram-positive cocci that are streps and staphs, with the exception of VRE and VRSA, are actually vancomycin sensitive. Now, these Abidotrophia granulocutellas will grow around the colonies of Staphylococcus aureus, and that makes them satellite. So here's an example of the Abidotrophia granulocutella, and you can see that we have a line of Staphylococcus right down the middle, and then that haze around that line is actually your organism satelliting around the Staphylococcus aureus. Enterococcus is another miscellaneous aerobic gram-positive cocci that is primarily opportunistic. It can actually lead to urinary tract infections and bacteremia. Now, this could be confused with the Streptococcus viridans group, or it may even be confused with Enterococcus, but usually they're more alpha hemolytic looking than gamma hemolytic. The gram stain revealed gram-positive cocci in tetrads. tetrads. Now, the Enterococcus can tolerate 6.5% salt, but don't confuse them with Enterococcus. They may be weakly catalase positive. Their PYR for Enterococcus is going to be positive, but the LAP, which is leucine aminopeptidase, is going to be negative. So Enterococcus viridans, like I said, is PYR positive and LAP negative. Enterococcus, on the other hand, are LAP positive. That's the main reason why we use the LAP test, to differentiate Enterococcus from Enterococcus. So Enterococcus is LAP negative and Enterococcus are LAP positive. Enterococcus viridans will tolerate 6.5% salt, but the bile esculin can be variable depending on the species. Gamella is another aerobic gram-positive cocci, and we do consider it a strep-like organism. They can actually be decolorized very easily, and they may have a flattened side and appear as though they look like a Neisseria species. So you want to be careful when you're staining it that you don't over-decolorize this organism. They're catalase negative, whereas Neisseria are catalase positive, and that's one way you can tell you're dealing with a Gamella and not a Neisseria species. Gamella are usually part of the normal upper respiratory flora and part of the gastrointestinal flora. It is rare, but they can be linked to some infectious diseases like wound infections and endocarditis. Now, depending on the species, you can have a number of variable test results, so we're not going to get into the specifics of Gamella right now, but do understand that they are all sensitive to vancomycin. If it's resistant to vancomycin, then it might be a Neisseria that you're dealing with, or it might be something like a VRE, but it's likely not to be Gamella if it is vancomycin resistant. Now, leuconostoc is very interesting because it's an aerobic gram-positive cocci that is resistant to vancomycin, and there are very few gram-positive cocci that are, but leuconostoc species is one that is resistant to vancomycin. They may appear somewhere between alpha and gamma hemolytic. It just sort of depends on the species. They're catalase negative, and they look like a strep and act like a strep, but it's actually vancomycin resistant, so we know we're not dealing with any typical streptococcus. They're a gram-positive cocci, and usually they are in chains resembling a streptococcus. It is considered to be an opportunistic organism causing bacteremia, respiratory diseases, abscesses, and those are usually seen in people that are already immunosuppressed, like people that have cancer or AIDS patients. So that's it. That's Unit 9. We talked about the gamma hemolytic streps, the Enterococcus, the Gamellas, the Enterococcus, the Abidotrophia granulocatellas, and the Enterococcus. Hope you enjoyed it.